Okay. Okay, welcome back, everyone. I think we'll start this session 7 on estuarine ecology, and I'll be chairing it this morning. And we'll follow the same format as we have been doing the last 2 days, where we'll ask each of the different presenters in this session to give a brief overview, 1 or 2 or 3 minutes on their specific talks, and just highlight their results from their studies. So I've divided the talks into about 3 different themes, and then we'll do the talks and then have a question and answer session. We'll bring in some YouTube comments if there is, or if you've dropped something in the chat, and then we'll go on to the next presenters. Okay, so first up today, a little bit outside maybe of the theme, and a leftover from one of the macroinvertebrate sessions on Monday, is in the lower reaches and in Durban. We have Jeffrey Lebepe talking about macroinvertebrates on a longitudinal gradient in the urban Palmit River. So Jeffrey, if you're there, you can go ahead.
Yeah, thanks, Vinand.
Yes.
I'm Jeffrey from the University of KwaZulu-Natal. So we looked at how the macroinvertebrates are responding to various land use, as well as physical habitat variation. So we hypothesized that since the river is comprised by various or highly variable stretches along the longitudinal gradient, there could be a variation with respect to community structure along the river system. And we also hypothesized that these macroinvertebrates communities will respond differently to different pollution level. And since this river was an urban stream, so we decided to include an anthropogenic litter as one of the components that drives this macroinvertebrates assemblage. So we hypothesized that in stretches where there is high accumulation of anthropogenic litter, there could be high habitat heterogeneity. So our results have shown that indeed there is a variation along the longitudinal gradient, which was highly associated with the pollution as well as the physical habitat. And for the stretches that were having poor habitat but with urban litter, it showed that this urban litter does provide some refuge for some macroinvertebrates. Thank you.
Okay, thanks, Jeffrey. Since it's the only one that's a little bit more on the freshwater side of things, if anyone has questions for Jeffrey from the audience, we can take it now. I think I had one question in your video, Jeffrey. You showed in one of the slides that that you found some amphipods or amphipoda in your system. So being in Durban and being, I assume, fairly close to the ocean, was those freshwater amphipods, or was it maybe some marine amphipods
They were actually freshwater amphipods. They were in the headwaters of this stream, so in the mountainside, which was bordered by urban habitat.
Okay, cool. Because that's not the amphipods are not always that common in freshwater systems, so it's nice record there for it.
Thank you.
Anyone else maybe with some comments? Okay, so if there's no comments directly now, we'll move on to the next presenter. But if you do have some comments later for Jeffrey, please feel free to ask it as well. Drop it in the chat box, and we'll ask it as we go along. So the second talk today is from Kezia Dreyer on the evaluation of carbon stocks and associated threats in the Burke River estuary. Kezia, are you there?
Hi, can you hear me?
Yes, we can hear you. You can go ahead.
Okay, so I'm Kezia and I'm from the University of the Western Cape, and my master's project focuses on blue carbon, which is basically carbon stocks found in coastal vegetated systems. And my study aims to quantify the blue carbon stocks in undisturbed and disturbed areas in salt marsh sites in the Burke River estuary in Valdrift. And I looked at 4 sediment cores in each site, and along with 4 biomass blocks. And I determined the sediment moisture content above and below ground biomass, as well as the dry bulk density by oven drying the sediment, and I determined the sediment organic matter by using the loss on ignition method, which includes oven drying the sediment at 550 degrees for 8 hours. And basically, my results showed that both above ground and below ground biomass was significantly higher in undisturbed sites, which was expected compared to disturbed sites. And in undisturbed sites, the organic matter decreased with depth, and in disturbed sites, the organic matter increased with depth. In conclusion, we saw a decrease in organic matter and living plant biomass in disturbed sites compared to undisturbed sites. And this means that disturbed sites have a great impact on the carbon stocks in the Burke River estuary, and management plans should be adjusted accordingly. Thank you.
Okay, thank you for that nice summary. Next up, we have Emily Whitfield talking about the comparison of primary producers as nutrient filters within an estuary. Emily, are you available?
Hi. Yes, thanks. My study had a look at how much nutrients, or namely the nitrogen, phosphorus, and carbon, 3 different primary producers can take up within an estuary. And we mostly looked at the phytoplankton, the salt marsh, and the seagrasses to see how much nutrients they can take up and store, and therefore act as a nutrient sink. So up until this point, my study is mostly focused on the literature, but for the phytoplankton, there wasn't much literature on covering this subject, so I have some preliminary results for that. And basically what I found from the literature and my own data is that salt marsh tends to have the highest uptake compared to the other 2 primary producers, so the seagrasses and the phytoplankton, and that the phytoplankton actually varies completely, and it can sometimes be a source and other times it can be a sink of nutrients, so it's highly inconsistent. And the importance of this is that eutrophication in estuaries typically causes salt marsh to reduce its below ground biomass, so it then erodes away and loses its ability to take up nutrients. And at the same time, the phytoplankton becomes more and more dominant because it takes up these nutrients very quickly. However, this is a problem because it can make our estuaries become a source of nutrients rather than taking up nutrients. And we need to manage this in order to reduce eutrophication, to reduce the amount of losses in our salt marsh, and to try and prevent phytoplankton blooms or harmful algal blooms, which can actually be detrimental to the ecosystem services. Thanks.
Yeah, thank you, Emily. And then next up, we will I'll ask Shwene Lakane to give us an overview on quantifying nutrient storage capacity of invasive aquatic plants in the upper SASAqS estuary. Shirley, are you there? Lakani?
I'm here. Can you hear me?
Yes, we can hear you. You can go ahead.
So I'm a master's candidate at Nelson Mandela University, and my study aims to quantify nutrient storage capacity of invasive alien aquatic plants at the SASAqS estuary. The main plant of concern is the water hyacinth, as it is the dominant invasive plant at the estuary and has been widely shown to store nutrients. And a huge bulk of my study is taking place at Perseverance, which is located at the upper SASAqS estuary. The reason for this is because Perseverance captures wastewater treatment, wastewater from all the 3 wastewater treatment works that discharge into the estuary. And so this will help us to determine how much nutrients all 3 wastewater treatment works collectively put into the estuary. The study duration is made up of sampling during low flow months, which is in winter, and high flow months, which are in spring. And flow is an important aspect of the study because water hyacinth are floating plants. Therefore, their cover at a site may change depending on the flow. So my study approach, at the sites, I'm collecting water samples as well as water hyacinth samples for nutrient analysis. I'm also using aerial images for the day of sampling to determine the water hyacinth cover in my study area. And this is achieved by mapping the water surface area of the study site and then adding the water hyacinth cover to the map to give a percentage of the cover. What I have noticed so far is that water hyacinth cover increases with increasing flow conditions in the estuary. And the plant nutrient concentration so far, the results so far from my first sampling period show that there is high concentration of total nitrogen compared to total phosphorus. And then the plants also store most of their nutrients in their stems compared to other parts of the plants. And to actually quantify the nutrient storage capacity of these plants, I will be using the water surface area, the plant cover, as well as the nutrient results to determine the nutrient concentration that will be stored by the plants on a large scale. And then using this information, I can then determine the possibility of using water hyacinth as nutrient filters in the SASAqS estuary. And my study is important in that there is a need to find solutions to mitigate the effects of increased nutrient inputs into the SASAqS estuary, as it is one of the most important ecosystems for maintaining biodiversity. It will also provide input on water quality management of an urbanized estuary that receives wastewater from wastewater treatment works. And then lastly, the study forms part of a larger research program on restoration of the SASAqS estuary that includes identifying innovative methods for water quality improvement. Thank you.
Thank you very much for that talk. And then the last one before we'll take questions is Tembela Vimbi talking about the role of disturbances in shaping microbial communities at 2 estuaries in the Eastern Cape. Tembela, are you available?
Yes, I am. Good morning, everyone. My name is Tembela Vimbi, a master's candidate at the University of the Western Cape. So my study titles the role of disturbances in shaping the microbial communities at the Wave Crest and the Nahoon estuaries in the Eastern Cape. So basically, like at the Wave Crest, it is situated in Transkei, which is part of the Eastern Cape that is not that well developed. So there's a lot of cattle browsing and communal farming and all of those type of things. So it's situated at a communal area. So it is mostly affected by cattle browsing. And the Nahoon estuary is located in East London, which is a developing area that is mainly disturbed by development practices and a lot of plastic pollution. So the study aims to determine the role of these disturbances in shaping the microbial communities at the Nahoon, Ngako, and Ngosi estuaries. This was done by assessing the impact of the main disturbances in each of these estuaries. At the Wavecrest estuaries, as I said, it is cattle browsing. This was done by firstly distinguishing between sites disturbed by cattle browsing and those that were not. In these different sites, soil samples were collected and 16S metabarcoding was used to investigate the differences in the microbial communities. And then at the Nahun estuary, we focused on the disturbance of waste input as plastics. This was done by measuring the environmental conditions at the Nahun estuary and also identifying the microbial community composition on the plastic bottles and the natural habitats of this estuary. So the main findings is that, firstly, in the site that is affected by cattle browsing, is that there's definitely a difference in the diversity and the composition of microbial communities in the sites that are disturbed by cattle browsing in comparison to sites that are not disturbed by cattle browsing. So there's changes in the composition and growth of microbial communities. Secondly, in the second site, the Nahun estuary, which is affected by pollution and plastics, there was a difference in the bacterial diversity between the insides of the bottles that we collected samples from and in comparison to microbial communities that were found in the natural habitat of that estuary. So that is it from me. Thank you.
Okay, thank you very much. Questions for the last four presenters, for Kezia, Emily, Shwene, and Tembela. I see there was a question for Tembela on YouTube saying, what future studies would you recommend in relation to the studies that you've done at present?
Okay, I will answer that by saying that firstly, in South Africa, there's not a lot of studies that are completed using the microbial communities and like investigating the DNA and such things. So the mangrove ecosystems are mainly studied through the investigation of the other disturbances. So it's very important for these studies to be like done. Also, like what I would do is mainly maybe changing some of the sampling methods and making them more accurate and more focused on getting, more focused on getting the specific conditions that will allow to test these disturbances accurately. Thanks.
Okay, thank you. There's a few questions and comments, Shwene, in the chat box saying, Lucian, do you mind asking, how does the spraying of glyphosate affect your plants, or are they always present in the SASAqS? And then a follow-up was, sort of, you can answer as well, if they act as such a nutrient sink, should we consider leaving them at the top of the estuary?
Can you please repeat the first question?
What does the spraying of glyphosate, how does it affect your plants? Or all of the invasive plants always present in SASAqS?
Oh, okay. So the invasive plants are always present at the upper reaches of the SASAqS estuary, and I'll answer the second part of the question by answering the
Hello?
Sorry. So leaving the water hyacinth in the estuary will be beneficial in that most activities such as fishing and recreation, as well as productivity and biodiversity, is concentrated in the middle to lower reaches of the estuary. So these reaches are of most concern, And also water hyacinth are freshwater invasives and are limited by salinity constraints. So at the start of my project, we found out that the water hyacinth were being sprayed with pesticides in summer by the Gumtoos Irrigation Board, but they had to stop doing that last year due to lack of funding. So they are already being left in the upper estuary. So we can take advantage of the ability of these plants to take up water and then use them as filters for the downstream estuary. What my study proposes is that instead of spraying the water hyacinth, we should keep them in the estuary until they reach their growth and nutrient storage capacity. And because they've been sprayed with pesticides, they can disintegrate in the estuary or in the downstream estuary and then take back the nutrients that they've stored. So instead of doing this, we can, once they reach a certain cover percentage, we can then commence with management control measures, such as manually removing them from the estuary to dispose them or be used in other ways. And this is because chemical control methods result in the nutrients that have been taken by the plants being taken back into the system. So alternatively, we can transpose them into water pools that have been created by sand quarrying along the estuary. These pools are not part of the estuary, and some of them have already been invaded by the water hyacinth. So I think, yeah, so yeah, that's that.
Thanks. There's another comment for you from Megan Reed saying, good talk, Shirene. I think it's important to consider various options for restoration for sure. You mentioned the weevils as biocontrol agents in your talk, and I wondered if you noticed the presence of these plant hoppers. Also, I wondered if perhaps it would be better to consider investigating and introducing native plants to the estuary for restoration, as opposed to using the invasive species to manage the nutrients.
Oh, okay. Yes, I think we can consider using native plants to do that, but the reason why we're using invasive plants is because they're already present in there already. So, and they already act as nutrient filters. And yes, we did find some weevil. Oh no, the weevil beetles, we haven't seen them yet because they haven't been controlling the water hyacinth in a while. So I haven't really seen them.
Okay.
So can I just make a suggestion for Enant and Trini?
Yes.
Yeah, please.
Thanks, Trini. Yeah, as I said, I think it's really important to consider various options. I'd suggest maybe, I really agree with what you were saying in terms of like reducing the use of chemicals, because obviously that just puts the nutrients back into the system. But perhaps it would be better to look, you know, like you'll see if you've watched my talk, or perhaps you watch later, I give a brief overview of the use of biocontrol to manage various invasive plants, including water hyacinth. So as you said, you're correct in saying like they're there and they're probably going to stay there, even if we use biocontrol, but perhaps releasing more biocontrol agents is something to look at instead of manually removing, to keep the populations under control. But I think maybe, and I'm not to criticize, more as an option to add perhaps in your thesis or something, is to look at inundating the system with natives rather, because we're seeing there's a transition where if we're not restoring the native vegetation, we have new invasives come in anyway. So I think it would be really cool to introduce that aspect into your research, perhaps just mentioning the use of natives as opposed to just the invasives. But yeah, I was impressed by your talk. I thought it was really good. I just wanted to add some input in that regard.
Oh yes, definitely. Thank you. I will surely consider that for my thesis.
Great.
Thanks, Megan. Maybe just a comment on that, also a question maybe on my side. I mean, it's good to use the biocontrol, but the nutrients still remain in the system once the plants die, and I think Emily alluded to it in her talk as well, where you can have mass release of nutrients if all of these invasive or natural plants release nutrients if they die off. And even with the biocontrol, you still sit with the extra nutrients, I guess, in the system that you would like to take out, preferably. Otherwise, you might have harmful algal blooms if the nutrients are released downstream into the estuary.
Oh yes,
Yeah.
that is why one of the management control was to manually remove them, because if they die back in the system, they'll definitely take back the nutrients they have stored back into the system.
There's a question in the chat for Kezia. Could you elaborate on the types of disturbances you found in your systems in the Burke River?
Hi, yes. The four main disturbances at the Burke River was erosion. Erosion mostly comes from the modified estuarine mouth that causes differences in the flows of the water and then causes more erosion along the banks. And then the second main disturbance would be farming. The salt marsh is grazed upon by many livestock, and farm vehicles and farm animals all contribute to disturbances caused by farming activities. Then there are a lot of walkways on the intertidal salt marsh that cause compaction, as well as driving on the salt marsh that causes compaction, and that is also a disturbance. And then lastly, there are lots of salt production sites, so salt pans that cause hypersalinity in the surrounding salt marsh, and all of these decrease the amount of carbon stocks in the estuary. Thank you.
Thanks, Kezia. I also had a question in terms of the carbon stocks, and if you looked at with the Burke River releasing ecological reserve flows from the Burke River Dam, and how that potentially could affect the carbon storage in the estuary. So the different sort of flood levels and water levels that's coming into the estuary.
I actually don't know, but what I can say is that at certain sites where erosion is taking place, there are higher water levels, and obviously the intertidal marsh would be drowning in areas like that. That's all I can say about that.
Any other questions for these four presenters for now? Okay. Does not look like there's any other questions, so we'll go on to the next four presenters. And first up, I would like to ask Phumza and Daleni to give an overview about the resource partitioning of 3 species of gobiidae in the Sundays estuary. Phumza, are you there?
Can you hear me? Yes,
we can hear you.
Okay. Good morning, good day, everyone. I'm Daleni Phumza from Rhodes University, and my talk is on understanding resource partitioning of gobiidae living in the warm temperate Sundays estuary. Okay, in my talk, I used different forms of resource partitioning in order to understand the coexistence of the 3 goby species that were dominant in the Sundays estuary. And thank you.
Okay, thank you, Phumza. Next up, we have Kylan Brown talking about the Cape Stumpnose in the SASAqS estuary.
Okay, hi, can you hear me?
Yes, you can go ahead.
Okay. Good morning, everyone. I'm Kylan Brown, a master's student from the University of the Western Cape, and my project looked at the diversity and abundance of epiphytic diatoms on the leaves of SASAqS in the SASAqS estuary. So the seagrass habitat is an important nursery area for many species of juvenile marine fishes, particularly for Rhabdosargus halubai, which is an abundant marine fish species found in estuaries along the southeast coast. And a study on the feeding habits of Aurolubi in the SASAqS estuary revealed that SASAqS dominated its diet. However, this species does not possess the digestive enzyme cellulase to digest plant material, so only the epiphytic diatoms and other algae on the macrophytes can be digested, while the macrophyte component passes through the gut undigested. And that's where my study comes in, as I aim to identify the epiphytic diatoms available for Aurolubi on the leaves of SASAqS, and thereby using diatoms as an indicator of feeding habitat. So it was found that the abundance of diatom species differed seasonally, with diversity showing no differences between seasons, and as no strong statistical relationships were observed between the diatom species and the environmental variables, it is suggested that biological factors such as the substrate characteristics and grazing may have been important in determining the seasonal variation in the abundance of diatom species. And species of diatoms that were present on the leaves of SASAqS in this study, as well as were observed in the diet of Aurolubi from the literature, included Grammatophora, Nitsia, Licmophora, Navicula, Coccinaeus, and Tabularia species. Thank you.
Thank you very much for that. It sounds like a really interesting study. Next up is Logan Lawrence, speaking about the distribution and abundance of microplastics in the Nahoon estuary. Logan?
Hi, can everyone hear me?
Yes.
Okay, cool. Good morning, everyone. My name is Logan Lawrence, and I'm a master's student at the University of the Western Cape. The project that I used for the presentation, its main focus was on microplastics. And I was given my interest in microplastics started back in undergrad, watching a documentary with on Carte Blanche. Then they were investigating the microplastic abundance in the different liquids that we consume on a day-to-day basis. And thankfully in honors, I was given this project, which is the abundance and distribution of microplastics in the Nahoon estuary. So microplastics data in the ocean is well documented. However, in coastal environments such as estuaries, the data there is quite scarce, which is what made my study so significant. So in order to quantify the microplastic abundances in the estuarine environment, it was vital that we identify the microplastic hotspots. And the Nahoon estuary is subjected to intense residential and urban development on the banks of the estuary, which results in an increase in human activities and interactions. I think it's also important to note that there's also a nature reserve as well as a boardwalk that's present in and around the estuary as well, which may also contribute to the interactions with the estuary. And many studies have indicated that estuaries located near development, much like the Nahoon estuary, are subjected to high levels of pollution. So therefore, the high disturbance levels of both plastics and development adjacent to the system was expected. And other factors, natural factors, include weather conditions such as rain and wind, as well as the state of the mouth that receives polluted water from the seawater, which adds to the overall pollution, which is why it has a total disturbance score of 16.5. And when compared to other estuaries like in Durban and KwaZulu-Natal, it's considered quite low. So the overall microplastic particles was 68,850 particles, in which surface water contained the most and sediment contained the least. And when compared to Govender et al. 2020, they found similar findings to what I found in my study, and they related this to seasonality. So I only sampled during the wet season, because I think I speak for everyone when I say COVID really limited us in 2020. So due to the fact that we only sampled in the wet season, we were only the amount of microplastics in surface water was quite larger than that in sediment. We also ran a linear regression test to determine whether there was a significant relationship between the nematophore density and microplastics. And we got a positive correlation, which also mirrors some of the findings in many other studies, such as Government et al. 2020. They also observed that Avicenna marina, which is also the mangrove species that was dominant in my estuary, was observed to trap macroplastics, which is why the nematophores might act as reservoirs for microplastics, which might cause the microplastic buildup in the surrounding sediment and water. So from the microplastics, we found 7 types of microplastics and 11 different colors. From the 7 types of microplastics, we found fibers, microbeads, film, fiber clumps, fragments, filament, and styrofoam. And from those 7, fibers and microbeads were the most abundant and contributed the most to the microplastic abundance. Fibers are believed to originate from ropes through recreational activities such as fishing, and microbeads are believed to come from cosmetic products. So from the 11 different colors, white was the most dominant in both sediment and surface water. And colors white, transparent, black, and blue were dominant in my estuary, which also mirrors the findings of many other studies. So just in closing, the varying types of microplastics and microplastic types and colors come from the diverse inputs into the estuary, and the degree of microplastic pollution in the Noon estuary appears to be a result of anthropogenic activities as well as natural factors. Thank you.
Thank you for that really interesting talk. And then for the last talk, we'll move to the rocky shores, and I'll ask Liam Connell to give us an overview of the upper thermal limits of 2 of our native limpet species on the southern and western coasts. Liam.
Thanks, Bernard. So this study formed part of my PhD, where we looked at the thermal limits of 2 limpet species along the South African coastlines, specifically looking at 3 marine protected areas, namely the Garden Route National Park along the southern coast, and the West Coast National Park and Namakwa National Park along the west coast. A non-invasive method was used to determine the real-time heart rates of these organisms, whereby thermal ramping took place under emerged conditions at a constant rate of 1 degree Celsius per 5 minutes. The thermal limits were determined using the Arrhenius breakpoint temperature for the sublethal thermal limits and the flatline temperature for the lethal thermal limits. So the aim of the study was then to determine the thermal limits of both species at all 3 sites, and to see whether there was any significant variation amongst these sites as well as between the 2 species. It was therefore hypothesized that Saphenara capensis would have a higher thermal limit across all 3 sites, and this was based on their physiological adaptations. So the results from the study showed that Saphenara capensis are better adapted to thermal increases, and this is under emerged conditions. And the reason for this is because they had overall higher sublethal and lethal thermal limits across all 3 sites. The reason Saphenara capensis were better adapted to the thermal increases, especially in emerged conditions, comes down to the physiological differences between the 2 species, where S. capensis have both a lung and secondary gills, whereas Scutellastra granularis have primary gills but no lung. And what these physiological differences mean between the 2 species is basically that S. capensis is better adapted to anaerobic respiration, and their respiration is more efficient during low tide, whereas Scutellastra granularis, on the other hand, don't have the ability to transport oxygen efficiently during the low tide or anaerobic conditions because they only possess gills and no lung. So yeah, that's just the gist of the study. Thank you.
Okay, thank you, Liam. So we can open up for questions now for the last 4 talks from Pumza, Kylan, Logan, and Liam. And I'll start it off with a question in the chat from Chantal Borense on why did you specifically look at the colored plastics and what is the significance of the color?
Okay.
Oh, sorry.
You can go ahead, Logan.
Sorry. So we wanted to have a look at the different characteristics of the microplastics, and those characteristics was the different types and the different colors. And the reason for that is because many studies have documented that these types and colors, they are related to the types of inputs of pollution into the estuary. So through looking at the different types, what was dominant, what was least dominant, we were able to determine, or well, not really determine, but kind of make an assumption on what disturbances were causing the different types of colors and types, or dominant types of colors and types into the estuary. I hope that answers the question.
Okay, there's a question from Pumza, and then I'll go to Emily.
I was wondering if were we asked to do the talk or just to summarize?
Just a summary, but
the real talk will come later.
It's all on YouTube, so everyone will
watch. Okay.
I think Emily, you have your hand is up.
Yes, I have a question for Carlin. I was wondering if you noticed if there was any variation in the diatoms, and if you maybe noticed if it was related to the presence of blooms in the water column.
Hi. We found there was a similar variety of species being found in each season. And my study took place in the lower reaches of the estuary, so there was regular input from the marine environment. So we didn't really take that into account.
Okay, thanks. I was just wondering.
There's another question for Logan on why microplastics would be higher in the wet season or show differing numbers with season, from, I think it was Kezia asking the question.
Yes, so the reason why there's a difference between, the reason why there's higher microplastics in the wet season is because the difference in the seasonality changes. So in another paper, Govender et al., they also documented that they had a higher abundance of microplastics in the surface water, and that's due to the rainfall that happens within the wet season, allowing the transport of microplastics from the sediment into the surface water. And they found differences in the dry season where sediment then had the highest amount of microplastics and surface water had the least. So I think it's safe to say that the microplastic abundance differs at spatial and temporal scales. Thank you.
It's just a comment from Lucia and Iman about that, on a follow-up on Emily's question on the algal blooms in the Swartkorps, and that these blooms are most in the upper reaches and the lower reaches are more marine dominated. Any questions, other questions you can ask anyone who's been in the session, if there's other questions you would like to ask? Richard Greenfield also is asking Liam what his significance of his results are in relation to climate change from the chat. Liam?
Thank you for that. So basically these results show that these organisms currently, what their thermal limits are and whether any temperature increases happen, especially with my study, we're looking at emerge. So if environmental sort of air temperatures increase significantly, will these organisms be able to cope? And whether they will or won't, it'll sort of have if they aren't able to cope, it will have a knock-on effect, because these organisms are both predator and prey, and so they will have less food to feed on, as well as there'll be less of abundance of them for predators to prey on.
Okay, thank you. Any other questions? I have another question for Logan. Did you look at, or maybe I missed it in the talk, on quantifying the input of microplastics from the river versus the ocean? Maybe the Nahoon is permanently open, I think, but in other types of estuaries where there's a bigger influence of a marine, is there marine or plastics that gets washed into estuaries?
So what we done was, starting off, the Nahoon estuary does receive a certain amount of polluted water from the marine environment because of the state of the mouth during different weather conditions. So yes, to answer that part of the question, there definitely is an input from the seawater, microplastics from the ocean. We haven't really focused on any river inputs. However, we have put together a sheet just to note all the disturbances, and then from there we gave it a score between 1 and 4, and that just gave us an overall total disturbance score to just see how disturbed the Nahoon estuary is. However, since this is the first microplastic data available for the estuary, I would definitely recommend that further studies have a look at the river input for the Nahoon estuary.
Yeah, thank you. Any other questions for the presenters in this session? Okay. A comment from Lucian here saying it's good to see such great estuary science being done and presented at this year's conference. Well done to all the speakers. And I can definitely concur with Lucian that SASAqS is always very well represented by estuarine scientists, and the standard of estuary science is always great. Another comment or question here for Kylan from Unrich saying, did you only study the diatoms on one species of seagrass?
Hi, yes, it was just on SASAqS.
Okay, thank you. Any final questions or comments for this session? If not, we can close it. Okay, it doesn't look like it, but if you do have any other questions for all of these presenters, please go drop them in the YouTube, and if the presenters can answer those questions in the next few weeks, that would be great. So then we'll close the session and we'll continue with the next session. On the program, it is scheduled for 11:35, but I think let's just start at 11:30. We'll take a 9-10 minute break, and then next up is e-flows of environmental flows, and chaired by Gordon O'Brien. So we'll see you in about 5 to 10 minutes. Thank you.